Okay, hi everyone. Welcome to this week's Machine Learning for Protein Engineering seminar series. This week, we are pleased and lucky to have Yeqing Lin and Minji Lee, both from Mohammed Al-Qureshi's lab. So Yeqing Lin is a PhD student at Al-Qureshi lab at Columbia University, and Minji Lee is an incoming PhD student also at the Al-Qureshi lab at Columbia. They will be presenting today Genie 2. So Yeqing and Minji, please take it away.
Thank you. Thank you very much for the introduction. Hi, everyone. So I'm Yeqing. I'm a PhD student in Mohammed's lab. So my work mainly revolve around like developing generative model to model the protein structural space and apply that for protein design. So today we'll be sharing our recent work on Genie 2, which kind of achieved state-of-art results in terms of like modeling protein structural space. And we are applying them to also like motif scaffolding task. Okay. So to start with, generative model and most recently diffusion-based model have like achieved substantial success in the field of computer vision, and they are capable of generating very impressive images with extremely high fidelity. And the main objective of diffusion model is to sort of capture the complex data distribution using predefined structures, for example, an isotropic Gaussian. And this will allow us to sample from the predefined distribution and map that to a sample in the data distribution. And one of the popular diffusion construction is the denoising diffusion probabilistic model that is proposed by Jonathan Ho. And the key concept is to gradually add noise into samples from the data distribution until it's completely indistinguishable from a sample from the isotropic Gaussian. And at the sampling stage, we could then sample from the isotropic Gaussian distribution and then gradually denoise it to generate samples from the data distribution. Given the success of diffusion model, this raises one question on how we could actually utilize this diffusion model to actually capture the complex protein space. And this would allow us to, by being able to model sort of the complex protein structure space, we are able to sort of draw sample from this space either conditionally and unconditionally, providing us an easier pathway to actually perform protein design task. So to apply diffusion model to capture protein structure space, we need to first understand like what a protein is. So essentially, proteins are one-dimensional polymers of amino acid that fold itself into a three-dimensional shape, or we call it structure. And this three-dimensional structure actually provides supports for various protein functions. For example, allowing binding towards a metal ion, or allowing interaction with small molecules or proteins. So based on this construction, two very common protein design paradigm will naturally arise based on this sequence structure function paradigm. So the first one would be sequence-based diffusion model, for example, EvoDiff, which attempts to capture the protein sequence space. And the main rationale is that sequences are actually ultimately the design target, and that given the availability of evolutionary scale sequence data compared to like existing structural database, that will provide us a lot more data and allow us to better capture the protein sequence space. Another very popular protein design paradigm is actually the structure-based diffusion, which aims to capture the protein structural space instead. So what motivates the development of this type of model is that structures are actually more closely related to protein functions. And by designing structures directly, it might be easier for us to actually design protein with target functions. So some of the models that fall into this paradigm includes our previous model, Genie, and also, sorry, the transition. Okay, some of them, for example, FrameFlow and also Chroma, and there are more to mention. And these models are sequence agnostic, and what they focus on is modeling the foldable protein structural space. And the generated sequence are then passed into inverse folding models, for example, protein NPMM, ESM inverse folding, to sort of obtain the sequences out of it. However, given the interconnectedness of sequence and structure, recently there are several attempts to sort of combine multiple modality into a single diffusion process. Sorry, the transition is not working correctly. Okay, and the first one to mention is called sequence condition diffusion in the protein structural space. So the idea is that we stem from the structural diffusion process, and the idea is to introduce sequence as condition into this structural diffusion process. This will allow generation to be conditioned on not just structure, but also sequence information. And this ability is kind of essential for many downstream protein design tasks, for example, motif scaffolding, because essentially what we want is the complete together with the sidechain arrangement of it. And one example to take this approach would be RF diffusion. And as we will talk in more detail, Genie 2 also fall into this category. Another approach to leverage both sequence and structural information would be to perform joint diffusion in the sequence and structural space, like what Multiflow did. And one other promising approach is to combine sequence and structure into a single latent space and perform diffusion in this latent space. And this is a very interesting direction, but to our best of knowledge, there isn't any existing model that achieve like competitive performance compared to the RF diffusion. So with understanding of different protein design paradigm, we will dive into more detail on sort of the development of our model, which is Genie, and then subsequently Genie 2, and demonstrate its superior performance in terms of capturing foldable protein structural space. So let's start with Genie. So basically, Genie is a structure-based protein diffusion model, so it doesn't involve around any sequence information. So it supports unconditional sampling from protein structure space, since it's basically modeling protein structure space, and would allow us to discover novel protein structures. And apart from that, by coupling it with a classifier or some form of differentiable loss function, we are able to guide this sampling process to allow us to perform downstream protein design task. So Genie consists of two main components, which the first one is the diffusion process with asymmetric protein representation, and the other one is an SE-free equivariant denoiser to sort of predict the noise injected at each diffusion step. So we'll go into that in more details. So to start with is a diffusion process with asymmetric protein representation. So Genie focuses on modeling foldable protein backbone structure, and particularly the C-alpha trace. So the reason that we model C-alpha trace instead of the full backbone atom is because that given the rigid structure of the protein backbone chain, we are able to reliably construct a full backbone atom given just the C-alpha traces using many existing bioinformatic tools. So focusing on just the C-alpha atom help us to significantly simplify the diffusion process without adversely impacting the model performance. So for the forward process here, we consider each C-alpha atom as a point. So we basically consider a protein as a point cloud. And then what we do is gradually inject Gaussian noise into the diffusion process, like we did in the original DDPM paper. And since the noise injected at each Gaussian step is small, the reverse process could be also approximated by a Gaussian distribution with the defined mean and standard deviation of variance. So here, the sigma theta here is actually the SE-free equivariant denoiser, which predicted the amount of noise get injected at each diffusion step. One thing to sort of point out is that in the reverse process, each protein is actually represented as a cloud of frame. So we see the F function there is actually a transformation function that transforms a point cloud towards a cloud of frames. And these frames are indeed just Frenet-Serrin frames. They are constructed based on consecutive C-alpha atom. So as an example shown on the left here. So this kind of provides additional orientation information for denoising and allow us to sort of leverage the expressibility of the SE-free equivariant denoiser and allow us to achieve better model performance. So in some sense, in summary here, our diffusion process is sort of asymmetric in terms of so our protein have asymmetric representation in terms of the forward and the reverse diffusion process to sort of maintain the simplicity of the diffusion process, while at the same time maintain sort of the expressibility of the model. And moving on, we will talk about the SE3 equivariant denoiser, which essentially is the epsilon theta here, which is the denoiser. So inspired by the AlphaFold2 architecture, this is kind of the architecture of Genie 2. So we are starting with the input reference frame, which is noised. So which is the at each time step is the reference frame time step t. So basically a featurizer generates and encodes per residual and residual pair features. For example, residual index encoding and pairwise distances from C alpha atom. And then these features are passed into the SE3 invariant encoder, which is the pink box below. So this encoder transforms these features into a single and pair representation and further refine this representation using triangular multiplicative updates. And given this latent representation, we then pass into the SE3 equivariant decoder, which combines latent representation together with the input reference frame to compute the updated frames. And finally, the noise are computed by taking the delta in the translation component between the input and the updated reference frames. And building on this architecture, we introduce Genie 2 with more components to sort of improve the model performance. So here for Genie 2, we introduces two things. So first part is a motif condition training, and second part is the data augmentation. So for the motif conditional training, it allows us the model to integrate sequence information into the diffusion process. And this would allow us to support motif scaffolding with one and also multiple functional motif here. So in this GIF, we show kind of one example of single motif scaffolding and another example of multi-motif scaffolding. So basically each motif has its own color code. And for the single motif scaffolding here, we notice that there's like two segments to a single motif. And for single motif scaffolding, the relative position and orientation between these two segments are passed to the model and are kept fixed throughout this process. And this is kind of different from sort of the multi-motif case, where sort of the relative position and orientation between these two motifs are flexible, which means that when we sample multiple samples, the orientation and position between them could be different. We will next discuss the motif condition diffusion process in more detail. So starting from the original Genie architecture, we start to introduce additional motif sequence information as kind of one-hot encoding into the per residual features, and additional motif structure information as a pairwise distance matrix into the residual pair information. The remaining architecture remain the same. And here we provide example of an input for a single and multi-motif scaffolding task. So for the single motif scaffolding task here, the intersegment distances is provided. So as we see in the first row, the distance information between segments are provided as templates, as a part of the structural templates to the model. But for the multi-motif scaffolding task, the intermotif distances information is actually not ablated out to allow like flexible position and orientation between different motifs. And here is the corresponding generated structures out of the model. And one key thing to sort of mention that is that there is a key differences between training of Genie and Genie 2, which is that while Genie is sort of trained on unconditional task, we train Genie 2 with single motif scaffolding task to give it a better model, since it gives us better model performance, which means that at each training step, what we do is we sample a structure from our data set, and then we generate a set of motif segments. Out of it, which are provided as motif to the model, and the model is required to sort of recover not just the motif information, not just the motif and scaffold, but the whole structures as a whole, but the whole protein as a whole. So here for the loss function, we use the same loss function as Genie, which is simply the mean squared error between the ground truth and the predicted noise. So there's no auxiliary loss here, but it's just the MSE loss. And this, by this construction, we kind of also break it down into the motif and scaffold part in our context here. And this sort of implies that the motif are actually sort of enforced as a soft constraint instead of a hard constraint, and allowing the model to sort of like be responsive to the motif specification while kind of designing the protein as a whole together. Also, another caveat to sort of mention here is that when we are training Genie 2, we does not train on multi-motif scaffolding task. So which means that we are only training on single motif task. And the reason is that our input specification sort of allow us to sort of underspecify this geometric relationship at inference time. So we kind of just train on single motif task and then using multi-motif scaffolding task as sort of like out of distribution generative measurement. Okay, moving on to the architectural change. Another thing that we find significantly helps with Genie 2's performance, to improve Genie's performance, is kind of the data augmentation part. So we perform significant data augmentation by using the recent AlphaFold protein structure database, which contains around 200 million structures. However, the issue here is like the database is pretty large, and then there are a lot of redundancy in terms of like structures there. So what we did is we applied the FOSIC. So we utilized the FOSIC clusters on AlphaFold database, which reduces the number of structures to around 2.3 million proteins. And further added to that is that not all the structures that are predicted by AlphaFold have very high confidence, which means that there is a wide range of like in terms of quality of the structure. So the way that we do here is like we further filters those by a PLDT threshold of 80, which leads to our final training data set of around 600,000 protein structures. And for comparison with like the PDB, so for PDB, the number of structures that will be available for training here will be around 20 structures. So we have like around 30 times more structures for us here. Okay, so that's all with kind of our architecture as well as model improvement. And before passing it to Minji to sort of discuss on our results, I also kind of discuss a set of metrics that we use for model evaluation here. Because unlike predictive tasks where the ground truth are actually available, you have a test set and a validation set to sort of compare against. There's actually no ground truth for generative tasks since we're kind of designing new structures here. So although the best assessment will be sort of an experimental validation, which we are currently working on with outside company on that, it is also kind of relatively costly to sort of generate these structures. So sort of to assess and compare the performance of Genie 2, we utilize this widely used in silicon evaluation pipeline to sort of like to access the performance. And this pipeline actually leverages on the recent development of both inverse folding model as well as protein structure predictions. So to start with, in this work, we compare Genie 2. We compare Genie 2 with both RF diffusion and chroma. So RF diffusion is kind of widely considered existing state-of-art protein structure diffusion model, and we are able to show that we are able to generate much more with equal designability and much higher diversity compared to RF diffusion. So basically, for each generative model, we sample a set of structure and structure and evaluate this set of structure based on a set of 4 metrics, which are designability, diversity, F1 score, and novelty. We'll discuss this metric in more details. So starting with designability, so how do we consider a generated structure as designable? So we apply the commonly used self-consistency pipeline. So the idea here is that we start with a So starting with the generative model, for example, a structure generated by Genie 2, we pass it through the inverse folding model, which we use protein ML in this case, which give us a sequence. We then pass the sequence through a protein structure prediction model, for example, ESM fold, and then we are able to get a designed structure out of it. And sort of to measure designability, we consider the consistency between our generative model and the desired structure. So basically, we compare the root mean square distance between them, which we call it SCRMSD, and also compute the TM score between them, which uses SCTM. So this kind of pipeline relies on the assumption that the inverse folding model and the folding model are performing good enough. And given sort of the resources that we have for like experimental validation, we think that this is kind of the best pipeline that we could use for accessing model capability for generative models. And for a structure to be considered designable, we use the commonly used threshold, which is SCRMSD less than 2 angstrom, which means that the root mean square distance between the generated and the designed structure is less than 2 angstroms. And the designability is finally the percentage of designable structure out of this generated set of structures. One issue to point out here is like designability doesn't measure the sense of it doesn't measure diversity, which means that it's possible that you achieve extremely high designability by just generating a single structure that's designable, and then over again, that will allow you to sort of achieve extremely high designability, but it could be misleading. That's why we are also using sort of the diversity metric here, which we think is a more reliable metric to sort of assess the performance of a generative model. So to start with, we have a set of generated structures. We first filter by designability and then filter out those that are not designable, which is the red dot. We then perform hierarchical clustering on these structures. So the distance between the cluster is actually measured using a single linkage method. So the TM score between the cluster is measured by the maximum TM score between any pair of samples between these two clusters. And we use a clustering threshold of 0.6, which makes sure that if we sample a pair of structures between clusters, the maximum TM score between them will be at most 0.6. So it will be less than that, which to be considered like unique cluster. So out of it, we consider the designability as sort of the percentage of the final unique cluster out of the total number of generated structures. Moving on for F1 score, we use the following formulation, which basically, if we substitute in beta equals to 1, which is the F1 score, the beta we are using, and then the percentage, the P structure and P cluster here is basically the percentage of structures that are designable, which is designability, and the percentage of unique clusters, which is diversity here. So this is kind of the final formulation of it, which means that we are just basically computing sort of the harmonic mean between the designability and diversity to give us a better sense like how the model balance between these two concepts. And finally, for novelty, we are using part of some of the similar stuff here. So essentially, to start with, we have a set of generated structure. We similarly filter out those that are not designable. We then compared it with respect to a reference data set. So here we compare to two reference data set. One is the PDB data set, and the other one is our training data set, which we call it AFDB here. So to sort of compute the distance, to sort of measure how novel a structure is, for each generated structure, we compare its distance against the reference data set, and the similarity is sort of measured by the equation below, which is just the maximum TM score to any structures in the reference data set. And we consider structures to be novel if the distance is less than 0, if the TM score is less than 0.5. And finally, we perform the similar strategy that we do in the diversity part. So basically do some form of clustering out of it, and then novelty will be computed by the final novel unique clusters out of the total number of generated structures. Yeah, with this, I will pass it to Minji, who will talk about the results part. Minji.
Yeah. Okay. Oh, thanks, Yeqing. Yeah. I'm Minji, and I'll continue for the results part of the Genie. So we evaluate Genie in three distinct schemes. First is unconditional generation. So mainly two analyses are done here. One is on in-distribution analysis that compares the overall designability, diversity, and novelty metrics discussed previously of Genie with other methods where the length of the protein is restricted to 256 residues. And this is the maximum length Genie has seen during training. The other is length-based analysis, in which we assess the trend of metrics by length and check how the models can extrapolate beyond the trained length range. And then multi-scaffolding aims to generate scaffold given the functional motif, such as enzyme active site. And lastly, multi-motif scaffolding aims to scaffold multiple motifs with unspecified intermotif positions or orientations. As Yeqing mentioned previously, this is achieved by not providing the intermotive information in the input structure template. And we created 6 tasks in our paper, and I will go through in the later slides. Yeah, next. This table shows the results of the in-distribution analysis of unconditional generation performance. So we generated 5 samples, each for the length from 50 to 256 residues. So we compare Genie with Chroma and RF diffusion, and frame flow is excluded in this table because it is only trained up to 128 residues, but it's compared in the later slide on the length-based analysis. And you can see that designability is as high as RF diffusion, while Genie is achieving much higher diversity and novelty compared to all the other methods. And adding to the diversity metric in this table, which is based on the global fold of the protein, we also analyze the secondary structure distribution. So here's the plots, and we compare the distribution of Chroma, RF diffusion, Genie, and AlphaFold database, which is our training data set. So while Genie can generate a diverse combination of secondary structure, it is biased toward helices. So we came up with several reasons for this. So first is that AlphaFold database itself is biased toward helices compared to other databases such as Copy or Swissprot. And helices are likely easier to generate than beta sheets as they involve local interactions, while sheets maybe involve long-range interactions. So these reasons might explain why Chroma, which employs a different architecture from Genie, is also biased toward helices. Yeah. And more specific to Genie, we suggest that low temperature sampling shifts Genie from the learned distribution. So you can see from the plot on the left that as the noise scale of Genie decreases, the density concentrates on the helix side. And in normal temperature, gamma equals to 1.0, the distribution of secondary structure is consistent with AFDB. So then, like, why low temperature sampling is necessary? In terms of performance, low temperature sampling is beneficial. So we use 0.6 noise scale for unconditional generation and 0.4 for motif scaffolding, as it gives better performance. As the plot on the left shows designability and diversity by noise scale. Then this leads to the next question: why does low temperature sampling gives us higher designability? So to investigate this, we ran our self-consistently pipeline on 1,000 randomly chosen structures from AFDB, our training data set, and PDB. So first, we observed that only 42% of AFDB is designable, which is only 42% gets self-consistency RMSD less than 2 and PLDT greater than 70. But using the same criteria, PDB structures, 87% of PDB structures are considered designable. But interesting observation is that using more relaxed criteria of desirability of SCTM greater than 0.5, 80% of AFDB is considered designable. So from this, we hypothesize that while FDB structures are globally reliable, local atomic details represented by SCRMSD, the self-consistent CRMSD, may be much less accurate. So we suggest that Genie can capture more designable space of protein structures with low temperature sampling. Yeah. I see some question in the chat, but like maybe we can address it after the slides. Yeah. Okay. Next, in the unconditional generation evaluation is length-based performance analysis, where we generated 100 samples for proteins of 50, 100, 200, 300, 400, and 500 residues. Despite having been trained on monomers of maximum of 256 residues, Genie can generate 3, 4, and 500 residue long structures with comparable or better performance than compared methods. For reference, RF diffusion in yellow on the plot uses a group size of 384 residues, and chroma in green on the plot includes protein longer than 500 residues in their training sets. So we'd like to share some observations on designability and diversity by the sequence length. So first of all, Genie is consistently generating highly designable structures while achieving higher or comparable diversity in all lengths. And the diversity of 100 and 200 residues long proteins is notably higher than competing masses. And for all masses, it is harder to generate longer proteins, showing a downward trend. And this trend is similar for diversity. It is expected that longer proteins exhibit higher diversity, but the diversity metric, as we defined, is limited by designability, as it calculates the number of clusters of only designable structures. So its maximum is limited by the designability. So that's why it's showing a decreasing trend. Yeah. So unconditional generation is all, and let's move on to the single motif scaffolding. So RF diffusion introduces 25 scaffolding problems on various functional motifs created from 6 publications. And we use those 25 problems minus 1 problem, which involves multiple chains, since currently Genie supports only monomers. And overall, Genie solves 23 out of 24 problems with a higher number of unique successes compared to RF diffusion. So in this plot, the y-axis scale is number of unique success in the log scale. So first we want to like clarify on this unique success. So previously people measured performance as the number of success, which is the number of designable proteins with accurate motif coordinates. However, this may be misleading since those structures may exhibit the same fold. In other words, the model may generate the same scaffold structure like over and over again. So we used a number of unique success as a metric. Similarly, in FrameFlow, the recent preprint on the application on motif scaffolding, which filters the unique structures by pairwise TM score comparison. So also the scale is log, so the performance gap is larger than represented in the plot. So the number of unique successes in the table, in the table might be would give a better feeling of the performance difference. So, and problem wise, as you can see in the plot left, this performance means that in 70% of programs, Genie is generating higher number of unique scaffold structures compared to RF diffusion. So these are some of successful scaffold designs of Genie, and the problem name is on the left. So here the motif segments are highlighted in blue. So you can see more of the examples in the paper. So, and also, as you can see, Genie can scaffold both continuous motifs and discontinuous motifs consisted of multiple segments. Okay, additionally, Genie solves problem 5WN9, where the motif is RSBG protein to the 10 sites, while RF diffusion does not. And Genie does fail on one problem, 4JHW, where the motif is RSVF protein size 0, that RF diffusion also fails on. We show 2 scaffold designs that are closest to the success criteria on the right. So here the three metrics, the self-consistency RMSD, PLDT, and PAE are within the success range, but the motif RMSD, which compares the design motif structure and the original motif structure, is slightly over the limit. So the RSBF side 0, the 4JHW problem, is a discontinuous motif with two segments, and one of these segments is less structured, which we speculate that it is harder for the model to design. And yeah, this is the reason why we're having the motif RMSD slightly higher than our criteria. Yeah. And finally, Genie keeps generating unique scaffold structures as the sampling size increase, and showing that it is capturing more diverse structure space. Yeah. So going to the next, we evaluate Genie in 6 newly created multi-motif scaffolding tasks, which we think have a large potential for complex protein design tasks, such as designing biosensors, signaling proteins, enzymes, and like protein-protein interaction disrupting or enhancing protein therapeutics. So in the previous single motif scaffolding problems, we had this continuous motifs consisting of multiple segments, but they are single functional unit, and thus the intermotif position is specified in the problem. So they are in a fixed orientation related to each other. However, to design proteins with new combinations of interaction partners or functions, it is necessary to scaffold multiple motifs without those intermotif positions specified. So this would give them flexibility in design and allow the model to find the best configuration for the multiple independent motifs. Yeah, so here is the summary of the benchmark. First is the scaffolding of two epitopes, each from RSBF and RSBG protein. And second and third are scaffolding ligand binding sites of the protein. And the difference between the two is how we split the motif. So the second one asks to scaffold two 4-helix bundles, each shown in red and blue on the structure on the right. But the next problem splits the bundle into two EF hand motifs, each binding to the ligand, and asks to scaffold 4 of those. And the fourth is scaffolding PD-1 binding motif, and fifth is scaffolding protein binding sites of IL-2 protein, as shown in the figure right. So the red structure is IL-2 protein, and the motif residues are indicated in different colors. And the last is scaffolding two 3-helix bundles acting as ligand binding sites. Yeah. Yeah, going on to next. Okay, Genie solves 4 problems out of the 6 created problems. And similar to the single motif problems, we report the number of unique success in this table. But like one caveat here is that currently we are not evaluating if one motif blocks the other motif, or if the scaffold is clashing with its interaction partner. For example, in the problem 2B5I, it has interaction partners. But currently it relies on manual checking since the structure number is a few. So, but like one future direction is including this as the evaluation pipeline. So we also show the successful designs for the fourth problem on the right, and for the problems 1PRW 2 and 4, the loops of the EF hand motifs that interact with the substrates are well exposed to the surface and are all unique successes. And the structures are diverse and clearly different from the original structure, with the motifs and the helix bundles distributed asymmetrically throughout the structure. So originally it was a symmetric structure with each bundle on the each side. Okay. And in problem 3NTN, Genie fails to scaffold two 3-helix bundles. And since the original structure was homotrimer, Genie fails to satisfy motif RMSD constraint for the motif which was originally on end of the chain. So in its design, Genie tries to connect like each segment of the bundle to the segment of the other bundle, and this connection distorts the motif structure and thus affecting the motif RMSD. So yeah, this can be one reason why it's failing in this problem. And we also speculate that the configuration of the motif and scaffold residues and the order might also be problematic. So it'll be interesting to revisit this problem with the future version of Genie trained to multimers also. And Genie also fails on the problem 2b5 by. In this case, satisfying self-consistency RMSD and PLDT constraints was hard. So only 5 structure passed this criterion, showing the complexity of the problem itself. Here we visualize the closest success with the motif RMSD and PAE slightly over the cutoff. Satisfying the motif RMSD constraint of the IL2 or alpha binding motif, shown blue, was the main complication. So configuration of the motifs. So yeah, so you can see on the blue, and you can see that this motif RMSD is slightly over the cutoff 1. So here we also think that configuration of motifs and scaffold lengths can also be the hurdle in this problem, since we didn't do much of the optimization on the configuration file. So we think it will be worth trying with longer scaffold lengths or like different order of configuration. And yes, this is also one direction we are trying to. Finally, we scaffolded the 3 RSBF epitopes as introduced in a recent preprint on multi-motif scaffolding with RF joint 2 in painting model, and the reference is in the below. And Genie solved this problem only with 1,000 samples. Yeah, and this is an example of our design. All epitopes are exposed to the surface and do not block each other, and also have a close structure with the original motifs, as you can see in the value of the motif RMSD. And finally, we are working on many directions to improve Genie to support more diverse downstream tasks with higher designability. So first is integrating multimer data to support binder design. And the next is we can also try on increasing the maximum sequence length, since currently it's 256. We are trying to increase to 512 or even more. And we are also working on improving the sampling scheme of Genie. And finally, we would like to thank to our advisor Mohammed and Professor Zhao Zhang, and also everyone from our AQ lab in the photo. And also thank you for hearing our talk. Thank you for your attention to this talk, and you can check our preprint in this link. The code and the checkpoint is also available in this GitHub link, and our context is here. And yeah, feel free to reach out if you have any questions or feedbacks. Thank you so much.
Great, thank you so much for that amazing talk. There are some questions in the chat, so I can just read them out for you. I think the first question was answered, so I'll move on to the second question from Alyssa. Is there an evaluation that demonstrates the improvements from augmenting the data set with AFDB structures?
So we haven't do the specific application out of it, but sort of compared to the performance of the original Genie, which is just trained on the scope dataset. So which is just trained on the scope dataset, which is also part of the PDB dataset, basically. So and we see a huge performance increase when we sort of augment the data with the AFDB, the AlphaFold database. So that kind of shows that the augmented data kind of give us a sort of substantial benefit there in terms of like performance. Yes, I think, does that answer your question?
Cool. I guess, yeah, also feel free to unmute. I'll check that. So yeah, also feel free to unmute to ask questions as well as leaving them in the chat. The next question is from Chen Wei. Will it be biased if you compare against the training data set, AFDB, between Genie and other deep learning methods which might not be trained with AFDB?
Okay. So the concept between like using experimental validated data like PDB or kind of generated data like AFDB is like much more complicated than that. So on the one hand, it's like AFDB does have like more structure compared to like PDB that's available. However, it's in another sense, it's that these AFDB structures are much more challenging to work with because of the quality of it. So I think in one of figure, we shows that if we using our own designability matrix using the SCRMSD, only like 40% something are actually designable. But if we're using the SCTM, around 80% of them are designable. So essentially structures in the AFDB, to us, it feels like the structure is pretty much correct, but there's a lot of refinement that need to be done in terms of like at least like locally. So it would be hard to say that is it a fair comparison? It is biased or not in some sense. Like we have more data, but the data is like much more noisy in some sense. So there's no clear answer here.
Great. Next question is from Olivia. I have trouble with this designability metric to evaluate binders or motifs generated for proteins from organisms that aren't well represented in the PDB. The PAE is usually very high, about 20. Others I've spoken with who are working with underrepresented organisms have noted the same. How could I overcome this and design confident structures with Genie 2?
Okay, so that might be a little bit challenging right now because essentially what a model learns to do is learning from the training data set. And if the sample is not like, at least like this underrepresented sample, if it's not present in the training data set, it might be harder for the model to sort of like actually capture or generate this kind of structures. So my one thought that I have is like probably the structures that you're looking for are maybe like less structures or contains like less secondary structures. So that might be the reason that they are harder to work with. So one possible way might be to actually to sort of curate this kind of this similar data set and then sort of fine tune Genie 2 on that. But I'm not sure like how difficult it is to curate this kind of like less order data set, and then how much structures are we able to curate out of it. So yeah, that's another interesting questions that we're also interesting about.
Cool, thanks.
Yeah.
Oh, yes, Minji, you answered the
Yeah,
next.
I just answered that one. Okay.
Okay, great. And the next one's from Simon. Could you speak to non-motif backbone RMSD between models, given that the provision of conditioning structures is provided as pair features rather than directly as in other networks?
Sorry, what do you mean by non-motif backbone RMSD? Do you mean in the training stage or in the sampling stage also?
Either one. So, right, like you do motif scaffolding.
Yeah.
And RF diffusion, for example, keeps the non-motif residues constant.
Yes.
So did you measure non-motif residue RMSD? Because your model effectively has to recover those coordinates.
Yes, correct. So at the training stage, we measure the noise vector for both motif residuals as well as scaffold motif residual, and they are given equal weight. So in some sense that we didn't fix the motif structure on its own. We provided with this information, but require the model to also generate it. So essentially motif is sort of enforced as a soft constraint there. And for sampling stage, it's more simple because we pass in the information there, the model generated, and what we do is some of the final checking to make sure that the motif constraints is actually satisfied.
Okay, but did you measure that in terms of like how hard is it for the model to recover those residues?
Not really. because we just kind of have a unified loss for the whole protein at the training stage, and we didn't kind of split it up.
Got it. Because you have that
Yeah.
plot of like motif RMSD versus SC RMSD, but that's a subregion.
Which I think that's for sampling, right? The motif RMSD there is the final generated one. Yeah. But for generation, we don't have the ground truth to compare, right? For generation, we have the motif ground truth that we want to generate, but we don't have the sort of scaffold ground truth. The model can generate whatever it wants, at least as long as it contains sort of the motif there. That's why we only plot like the motif RMSD with the SE RMSD.
Got it. Okay, I think that maybe I got the terms backwards. So you're calling what you're generating not the motif.
Yes.
Okay.
Yeah.
Cool. Next question from Sean. Is there any amount of wet lab validation ongoing for the successful solutions to these design problems? Perhaps not feasible, but for something small, globular, and soluble could certainly be crystallized to show that these designs work in practice.
Yes, we are currently working with our external partner to sort of like at our current stage, we are testing on some of the RSVF design that we are generating, both containing like 1 motif, 2 motif, or even 3 motif to test like the stability as well as the effectiveness of them. So we are currently working on that right now.
Very awesome. Shu Hao asks, how is the motif encoded?
Okay, so the motif contains 2 parts of information. One is the sequence information, which is encoded as one hot encoding of each residual type, and then get kind of concatenated to the per residual feature vector there. And then for the structure, we kind of represent it as a pairwise distant matrix between the C alpha atom of the motif. And then that kind of give you a sort of distant 2D distant matrix, and then kind of concatenated as part of the residual pair features there, and then get passed into the model.
Great, awesome. I think that's all the questions in the chat for now. Any more questions from the audience? Okay, if there are no more questions, thank you again to Yeqing and Minji for giving that great talk.
Yeah, thank you everyone. Thank you.
you.
Yeah, feel free to reach out to us if you have any like after thought or any questions that you want to like, or comments that you want to give us. We appreciate your attention here.
All right,
Thank you
great.
very much.
Thanks everyone.
Yeah, bye.
Bye. Thank you, everyone.